All right, this module is going to shift our discussions of adaptations from environments to infectious disease, and we are going to start with talking about malaria. Now, malaria, P. falciparum, is a parasitic protozoan that infects human red blood cells via transmission from host mosquitoes. Within the red blood cells, the protozoan reproduces, and this process results in the destruction of the host cell and dispersal of protozoans into the circulatory system. All right, symptoms include initially fever, nausea, and body aches, but can lead to severe symptoms such as anemia, hypoglycemia, cardiovascular collapse, low blood pressure, and kidney failure. And often, malaria results in death. In 2013, the World Health Organization reported 198 million cases and totaled 500,000 deaths. Majority of deaths were of children located in the African continent. So, a brief historical overview of malaria. So, human populations had limited encounters with malaria until about 10,000 years ago. The development of agriculture brought about ideal conditions for the malaria parasite. Cleared land provided locations for stagnant water collection necessary for mosquito breeding. Increased human sedentarism and population size increased the likelihood of humans being host to the malaria parasites. Malaria since that time has exhibited a strong selective pressure on human populations. So in order to really look at malaria, we need to understand a bit about hemoglobin and a disease called sickle cell anemia. So hemoglobin is made up of 4 polypeptide chains, 2 alpha subunits, and 2 beta subunits, and it is responsible for the transport of oxygen. Now, sickle cell anemia is caused by a sickle cell gene. This gene is a mutation caused by an SMP in the hemoglobin A gene. Homozygous individuals with SS have sickle cell anemia, and they typically die at an early age before reaching maturity. Heterozygous individuals, AS, have a mixed phenotype in that they have sickled and normal cells, which results in them being physiologically normal. Now, there are several explanations for how sickle cell anemia originated. So there is the single mutation theory, and this is that the S allele existed in low frequencies in African populations. Upon expansion into South Africa, in conjunction with the adoption of Swindon agriculture, resulted in an increase in malarial infections. This increased the frequency of the S allele in the population. Gene flow then allowed the spread of the S allele to other populations, being India and Arabia, etc. Now, the other hypothesis is what is called the multiple mutation theory. And so modern genetic studies indicate that the S allele arose multiple times in different populations, indicated by different DNA polymorphisms found in different parts of Africa and Asia. The polymorphisms were named for the original discovery location, include variations of Senegal, Benin, Bantu, Cameroon, and Asian forms. Now, there are some complications with the multiple mutation theory. So, during meiosis, gene recombination can occur, which in some instances results in gene conversion, which is a process of inserting a gene or a portion of the gene into another gene. So in African populations, nearby different polymorphisms may have developed. When these populations experience gene flow, i.e. interbreeding, the S allele may have been transported into the divergent polymorphisms. The takeaway is that the S allele may have developed only once, and the different polymorphisms may be a false positive. Now, sickle cell anemia as a disease is a homozygous condition that results in chronic anemia and often results in premature death. As such, it is expected that such fatal disease would be eliminated from a population via natural selection. In many populations, this is true. However, in some, the frequencies of sickle cell allele are quite high, up to 30 to 35%.